Good morning. It's our great pleasure to have two speakers in the first section. The first one is Dr. Sean Greenman. He's the director of the Center for Cancer Research in University of Melbourne. Dr. Greenman received his PhD degree in human cancer genetics from University of Queensland, and he received two scientific fellowship training. The first one is the Royal College of Pathologists in Australia. The second one is the Australian Academy of Health and Medical Sciences. Over the last 20 years, he is the pioneer in dissecting the underlying genetic event driving the pathogenic state through the multi-omic analysis. And his topic today is the cancer of the unmet need and the precision oncology. Let's welcome Dr. Greenman.
Thank you very much for the Thank you very much for that very warm introduction. I begin by thanking Professor Simon's colleagues for the opportunity to come back to Taipei. It's been 9 years since I've been here. It's a pleasure to be back again. I very much enjoyed the meeting yesterday. I think some of the work done yesterday was very exciting indeed, and very important to see that outreach taking place, and very much looking forward to what we're going to see today. What I'm planning to do with today's presentation is give you an introduction to our experiences on building the path for testing precision oncology within Australia, and really working out how we prepare to tackle some of the most challenging cancer patients that we're seeing within our hospitals, where we believe that genomics will play a key role in improving their outcomes. So I guess in terms of the beginning of the path towards precision oncology for us in Australia, it certainly started with the likes of the International Cancer Genome Consortium. This was a global effort which we took part on. We started in 2009, and in 2010 we in fact published our first patient's whole genome transcriptome data. There were 4 pancreatic cancer patients that we described. One of the unusual things about the Australian program or contribution to ACGC is that for the majority of our programs that we undertook, the patients were collected prospectively. We did not take them out of the research samples in a freezer. They were prospective patients, and they were consented so that if we found things that were likely to impact upon their treatment or indeed relate to predisposition of cancer within their family, the patients had agreed to have that returned back to their clinicians. So we've actually had a long time to fall over, many times, to work out how to optimise the systems whereby we should be able to do that within our community. A second thing we've seen in recent years is the federal government funding an initiative known as Australian Genomics. And this is an interesting program that, rather than generating a large amount of genomic data, it's actually invested in building the networks, in developing the government policy, in establishing the links internationally so that we can share data between countries, bringing the community together so that we have the advocates for rare disease matched up to the diagnostic labs and the clinical groups all working together. And I think we saw yesterday very good examples of how all of the way that we treat patients today involves expert teams all coming together. And this has really been a way that we've been building those networks so that if we go to the next level, we've actually got the workforce and the know-how and the frameworks to do so. And then I guess if we go more recently still, I'm very excited to say that our government has just recently announced that they recognise that the time is right to take the next step in advancing the use of genomics in healthcare, and have announced a $500 million Australian initiative which is due to commence sometime this year, and use that to promote genomic research, to really drive studies to show where genomics can improve outcomes for patients. And indeed the areas that they're interested in pursuing are the likes of rare genetic disease and indeed cancer. We actually, shortly after this was announced, we had an election, We all expect to see a change of government. I think even the government expected to see a change of government. They've recently been re-elected, so now they're trying to work out what are their specific goals going forward. But I think it's fair to say for us it's an exciting time, and we're looking forward to see how we can make a meaningful change in the years to come. I guess one of the things that, when thinking about this sort of opportunity and where we might want to take our research, we've recognised from our discovery-based approaches that there are definite opportunities that we can pursue when it comes to cancer patients, where we think we can show clinical utility quickly, and indeed we think we can have the best chance of making a meaningful improvement of outcome for patients. And I'll describe those sort of areas strategically that we thought were key places to go right now. So the first thing I want to highlight is that it is well recognised both within our government and our community that the experience for cancer patients differs wildly, and there is a real inequity in the types of outcome patients can expect. And the graph I'm showing you here is Australian data, which shows you the percentage change of 5-year survival between the 1980s and mid-2000 or 2013. And indeed, the good news is that the arrow in the middle is showing you that since the 1980s, 5-year survival for all cancers overall has gone from 45% to 70%. So that's really quite a dramatic change. And you can see some real success stories like prostate cancer or breast cancer, where they've now gone up to 90% or thereabouts 5-year survival. So overall, we're actually being traditional oncology and traditional trial approaches have made real progress. The challenge is that there are many cancer types that are being left behind. And so the cancer types in the top left-hand corner are those that have not seen that benefit. And indeed, all of these have less than 20% survival over a 5-year period. And given the small size of those arrows, you can see that in fact there hasn't been much change since the 1980s. And if we go through and have a look at that particular group of tumours up there, we actually end up seeing those and the rare ones that really aren't listed on here. About a third of our fatalities each year are in that top left-hand corner. So in terms of cancer deaths, we're really struggling with this group, that group in the far left-hand side. And if we dig down and have a look at those a little deeper, we start to find they have some similar properties. We have those that are considered recalcitrant. These are cancers that are resistant to most of the drugs and developments we've seen that have benefited other solid tumour types. They can be enigmatic, and by that I mean the microscope fails. When we look at them down the scope, or we use traditional pathology, they're a conundrum. They're unclear as to where that tumour may have come from, and if we don't understand that, we can't even rely on traditional oncology. And then indeed we have cancers there that are rare. We have never developed a playbook, so we don't know, out of all the drugs that are out there, we haven't done robust randomised trials because there aren't enough patients there to actually teach us those lessons. I guess the other thing I want to highlight about inequity is even if we go through and look at one of these cancers, say cervical cancer, where we've seen that survival overall isn't too bad, the reality is if you dig down and look into the various histological subtypes that we see within there, some of them do much poorer than others. So even within what we think to be success stories, there may well be quite malignant subtypes that still are being left behind because overall the stats are looking pretty good for that tumour. And then finally, the other thing I want to say is that this graph here represents the experiences of large groups of people, and certainly what everyone in this community would be aware of is that when we get down to treating a patient, it's about treating an individual. All of our genetics is different. And indeed, within each type, we can see patients who do very, very poorly, and the standard of care does not work, either because that patient can present quite late rather than quite early. That patient may originally be very sensitive to a therapy and then acquire resistance. And once you've done that and run out of opportunities, you're left behind. And then finally, we have those patients that we call outliers. And these are the patients that present with what you would typically think to be an indolent disease. You will treat with curative intent. And despite the fact you expect that patient taking years if you're going to lose them, you may find that their cancer is far more aggressive than you'd expect and far more resistant to the drugs that you would traditionally use. And at the moment, these patients, they are a challenge, and they're patients that are left behind. So with that in mind, we've been interested in commencing a program within my centre, the University of Melbourne Centre for Cancer Research, where we're focusing on these cancers of unmet need. So these are the cancers that we feel the patients are being left behind by the standard of care. And we're really drawing on a network of hospitals which sit around this circle here on the outside, and in the middle of that network is a hub called the Victorian Comprehensive Cancer Centre. That was the beautiful building I showed you on my introductory slide. It's a very large cancer-specific hospital with a research hub built on top, and that is where I'm currently located. And we've been interested there in rolling out a platform for genomic pathology, and then coupling that with a clinical trials network that spreads across this hospital group to actually go through and systematically test these approaches in challenging cancers. And then just a little map of Australia on the left-hand side. The dark blue component is the state of Victoria. That is where I'm currently located. And indeed, all these hospitals sit within that state. Our healthcare is primarily public healthcare, but we deliver our healthcare state by state. So each state has it, similar to Canada, each province or state has its own system to deliver that care. So collectively, this network can capture about 20% of the Australian population, and every hospital I'm referring to here is a public hospital. And then I should also highlight that this initiative includes a number of our leading research institutions, namely the Walter Eliza Hall, University of Melbourne, the Peter Mac, and that actually brings both our translational knowledge and this clinical network together. So the VCCC, the platform that we've established there, is very similar to the pictures that we'll see from many other initiatives being set up globally. We have developed a protocol for identifying patients. We have a group of clinical experts that will agree that a patient is appropriate to be included. We have informed consent, and then those samples are passed over to the laboratory. We have two processes available. One is panel-based, one is whole genome transcriptome-based, and then indeed those data are now being turned out in a standard fashion. They're then being turned over to a molecular tumour board, where we have representatives from that hospital network who will then weigh that information with a multidisciplinary team to make recommendations about how to go forward. This is a prospective study. We're collecting these patients in real time. And in the first instance here, the first phase has really been feasibility. It's been about educating our pathologists and our surgeons and our oncologists and our bioinformaticians to talk together. That might sound like a straightforward thing to do, but there's a number of different cultures there we've had to work through. And then indeed now what we're looking to do is there's an expansion of an innovative trials network that will run with this so that we can start to, when we find vulnerabilities, make the most of those in our patients. All right, so there is an age-old dilemma, older than time itself, I think, when it comes to cancer genomics, and that is what sort of assay to use. And as you saw, we have the ability to use a panel, a comprehensive panel with about 400 genes, or indeed go and carry out the likes of whole genome transcriptome sequencing together. And collectively, the community has come down with the current rationale that where there are well-defined cancers and there is a drug for which it is matched to a specific mutation, and we know the overall biology of that cancer isn't too complicated, they'll pursue a panel-based approach to have a look at what is going on within those cancers. If, however, a cancer is more challenging, if it is something whereby the pathology is ambiguous, if it is a cancer for which we know that the types of mutations that can drive that cancer, drive that transformation, aren't point mutation based, we will think about using the whole genome approach. We also recognise that the hard to treat cancers, we want to be able to understand everything that's going on in those to see if there's any weakness we can exploit. And then finally, there are some cancers where prognosis is unclear, and certainly in the likes of the hematological malignancies. Understanding prognosis is key between making a decision between transplant and treatment. And in fact, we saw a very elegant presentation yesterday describing that in leukemias. So these are the sorts of areas where we're looking to take the whole genome transcriptome approach to. And that's what I'll focus on for the rest of the talk. So why whole genome based approaches? Well, I guess I fall back on our experiences within IGC, and I can give a whole lecture as to why we should be doing that rather than panel. But I just want to highlight some of the areas that have been very important to us. The first one is, it's not just about point mutations. The graph here, the plot you can see, I just want you to take note of the red events, which are inactivating events in well-known driver genes in high-grade serous ovarian cancer. When TCGA first presented their results using exomes, they missed all of those events, and they missed all these drivers. Now, many of those drivers are actually druggable. And indeed, without going through and using a whole genome approach, they were invisible to those events. Many of them are copy number neutral. They're a rearrangement of the genome, which leads to exons being flipped over. The gene is ruined, but by exome it looks completely normal. So in order to capture these events, which make up about 50% of the key driver events within high-grade serous ovarian, you must use whole genome sequencing. We've also seen other examples of where rearrangements can bring an enhancer and put it next to a gene to drive the activity of an oncogene. And in fact, there are whole classes now of paediatric brain cancers which are defined by these enhancer hijacking events. And so without whole genome, you can't actually detect those because these events are an opportunistic enhancer being put next to an oncogene. So it's a very big search space to go looking for. You really need whole genome to make sense of that. A very surprising finding we found in esophageal adenocarcinoma was that approximately 50% of the tumours that we looked at showed that they'd undergone a genomic catastrophe, where one or two chromosomes were being smashed into a million pieces, and indeed esophageal adenocarcinoma will glue many of those pieces back together, and if they can stabilise with an oncogene, that becomes the dominant oncogene driving that tumour. Now, one of the challenges with that is that this means that different esophageal adenocarcinomas can opportunistically pick up different oncogenes. So it's not just KRAS driving that disease, it can be many, many different oncogenes. And if you think about a precision medicine approach, you must be able to identify that sort of phenomena in order to think about targeting the disease. Finally, we've seen some really elegant work coming out of OICR showing that in the early stages of pancreatic cancer, it's possible that in one cell division, about 15–20% of the patients will see a catastrophe, which can lead to the destruction of multiple key driver genes in a single cell, and you can go from pre-malignancy to malignancy very, very quickly. The events that I've described to you aren't necessarily rare in those cancer types, and in some of these they are the dominant process for causing mutations. In others it's a sizable percentage, but really you must think about, if we want to make sense of patients, one patient at a time, these challenging patients, we need to be able to survey and include or exclude these events. Finally, I should also say there are other sorts of evidence talking about the promoter mutations or other sort of enhancer-specific phenomena. We're now finding that these things identified in one cancer spread across many others. So we have to be able to survey all these events equally. The second thing I'd like to highlight from whole genome sequencing is the importance of understanding the global pattern of damage. And in fact, we heard yesterday with some presentations about you can look at the global somatic pattern of damage in tumours. Here we're not looking at genes that are mutated, we're just looking at the pattern of damage. And indeed, the patterns are quite distinct here. We don't need to go through the details of what they exactly are, but these are global patterns of somatic damage. And they can be tied back to particular mutagenic processes or deficiencies in DNA damage repair, and we can use those as a diagnostic within that individual to recognise those deficiencies. And indeed, this sort of approach tells us about the mechanism, but given that different cancer types have different patterns of these sorts of things, we can start to use this information to work out where a tumour might have come from. So with that in tow, we've actually gone through and established a platform that will systematically review whole genome and whole transcriptome and then ultimately put that data together. And that when we go to write a report for our patients, these are the sorts of steps that we review, and I'll walk through these to show you that the genome and transcriptome together is a very rich combination. I should say that the platform that we use here has been built by Oliver Hoffman, who leads our informatics. It's largely based on BC Bio platform. It's currently running on HPC. It's also been implemented in the cloud. We've been very actively involved with the Global Alliance looking at how we make these sorts of systems interoperable, and indeed actively working with Illumina to see how we can start to integrate these sorts of efforts with their cloud-based approach. So if we think about these pipelines, all pipelines want to make sense of what's happening in the germline and what's happening somatically in terms of rearrangements. We can go through and summarise those globally for a patient where we're identifying all the mutations. We're looking at the likes of where the copy number is, where there are gains or where there have been losses. We will also look at the rearrangements, trying to understand the overall stability of that. And that's a standard sort of output now that we can turn out on our patients. We also, once we have that, there's a number of things around the patterns of damage that we'll make readouts on. And these really give us insights into, is there an obvious signature of a DNA damage repair deficiency, or is there a dominant mutational mechanism there that may give us insights about where the tumour came from? But we can also assay things like microsatellite instability, or even the telomere length, which can be useful in diagnostics, certainly in neuroendocrine tumours. We have a system now for going through that. Once we have all of those events, we're also able to now start to annotate those with key information, trying to understand what the biological consequences of those. We use a variety of tools that are open source. We're actively collaborating now with Purion to see how we can take these sort of whole genome data, that being copy number, structural rearrangement, and point mutation, and integrate that into their system based on the outputs that we have here. And this sort of information can be useful for biologists, but really we must tune it so that it's going to be applicable to our clinical colleagues. The next thing we've got is that's the DNA side of things. What about the RNA side of things? How are we using that? Well, currently we analyse the DNA and then we analyse the RNA and we put those together. And we use the RNA to build evidence that we think when we see events in the genome, we give confidence that we believe that they are real. So examples of those are included as follows. If we have a gene that has a particular mutation, we can go through and then have a look at the relative expression of that individual. So this is the black dot versus where we, this is the distribution expression of about 400 pancreatic cancer patients. So we're trying to understand where that we've actually analysed in-house. We can then actually have a look at where they sit in the distribution of that cohort. So it's showing those that with the mutation actually have a very low level of expression compared to the other colleagues within the cohort that we're looking at. When we find the likes of splice site mutations, we can immediately now start to look at evidence or supporting evidence of intron retention. And when we find the likes of oncogenic fusions, this here is a RAF1 fusion seen in a pancreatic acinar tumour, we can actually have a look at the overall expression of the active oncogenic component of that gene, showing that it is much highly expressed compared to its peers. So it's a way to start to validate those. We can also look for the presence of the oncogenic fusion between the partners that we're seeing the DNA in the RNA. As we've been hearing, immunotherapies are of increasing importance, and there is a dilemma there around the ability to understand patients that will respond well or patients that are likely to be resistant. And that is another thing that I could talk about, a whole different talk. But we have an increasing number of tools now which we've been using to make sense of the quality of neoantigens that patients may have from their somatic mutations. And indeed, we've shown recently with colleagues at MSK that the quality of your neoantigens can significantly, even in pancreatic cancer, be protective for a small group of individuals when they are of high quality, which is very different to the standard of outcome that patients have. So this is, I guess, another toolkit that we don't routinely use, but certainly we've integrated into our systems. And then finally, we have a phenomenon called tissue of origin. And this is something that I think in TCGA and ICGC we recognised early on. And we just thought that sometimes this was a quirk of our research activities. Moving into a diagnostic setting, we've actually found that about 4% of the samples that we see coming through in our challenging patients, the expected tissue of origin for that tumour is not what the oncologist or indeed the pathologist was expecting. So tissue of origin is where you see something like you find a pancreatic cancer tumour, and there's a mass there, they take a biopsy, we go and sequence it, and then we find that it has a somatic UV mutation signature. So those cells must have had some exposure. And then indeed, if we go and look at the expression of that tumour, although it may have been dedifferentiated and not particularly useful to the pathologist, the expression can start to tell you that it's expressing key transcription factors that are seen in the likes of the skin rather than what you're actually seeing in pancreatic tumour. So we can start to flag then that this may be a metastatic lesion that has moved to the pancreas rather than just a dedifferentiated pancreatic tumour. And if you think about it, when it comes to tissue of origin, we can use gene expression and we can use patterns of damage to get insights into that. So indeed, the plot at the top is just showing you a UMAP. It's a way of graphing global expression of 10,000 samples from TCGA, where we've isolated the genes that we believe demarcate tissue of origin, and then we can in fact project a patient onto this. And in fact, there's a little dot up here which represents a cancer of unknown primary that's sitting on top of, in this case, a large group of breast cancer samples. So the expression alone can actually improve that resolution, where in this case, standard histology and histopathology couldn't actually get us any further. There are activities, and I draw attention here to, once again, colleagues from OECR, which have been using machine-based learning approaches to look at global patterns of whole genome sequencing data, and in doing so, being able to predict where a tumour came from just by its pattern of somatic damage to an accuracy of about 85 to 90%. And then finally, we can use these sorts of signatures that if you find a pancreatic tumour with a smoking signature or with a UV signature, we know that that tumour couldn't be physically exposed to those mutagens, so it must have come from another site. So all of those are now built into any sample that we read through now, trying to make sense of where it comes from. Finally, pathogen detection. This becomes very important for some cancers where we know there is a viral contribution, certainly our gynecological work. So we've worked up the approaches there to be able to screen the unmapped reads for viruses, and then that process to work out where they map to understand the consequences of oncogenesis driven by those particular viral components. So we can identify both where the virus may be located within the genome and indeed the consequential areas that it may be amplifying in a particular patient. The sample I'm showing you up here, I draw your attention to, we can see where the virus is integrated. There are two sets of well-known oncogenic genes actively being expressed here, but as a consequence of it being inserted into the genome, it's amplified the MYC as a super enhancer without amplifying MYC. And as a consequence, this drives a massive level of MYC expression, but by panel, this patient had no observable damage to the MYC gene, so it was presumed to, the key driver profile was completely different. Finally, when you've got gene expression by itself, there's a number of other things we can do with that. These are very much research assays, but we're trying to ruggedise those, where we're going to use the pattern of gene expression in key gene sets to make sense of things like immunomodulation and indeed the activity of DNA damage repair. And if we do these robustly with our other pathology tests, we have a chance now of bringing these in to be a biomarker that we may be able to use in the next phase of our activities. So just in the last 10 minutes, what I'd like to do is give you some experiences of what we're seeing with some of the key themes within the cohort at the moment, when we've taken this whole genome transcriptome approach. So you will have heard that the pancreatic cancer is something I've worked on for quite a while. It's quite dear to my heart. The statistics really in the graph on the left say it all. That is the overall survival post-diagnosis in Australia, whether you have localised disease or whether you have advanced disease and diagnosis. Median survival is about 7 months. It actually is a major burden of cancer mortality within Australia, and unfortunately the rate of pancreatic cancer, certainly across the Western world, well actually across the world now, is steadily increasing. It is now the third most common cause of death in the US, cancer-wise, and is expected to overtake colorectal cancer in the next 5 years. So this is clearly a cancer that is recalcitrant, and we've been keen to see how we might be able to dissect this particular disease using genomics and see if we can improve outcome. I should say that the way that we collect our patients, given how challenging it is to tackle this disease, and given that we still don't have enough drugs that actually show a positive signal, we have an activity whereby we are carrying out our genomic pathology on patients and able to return information. But there is a sizable activity where we're generating matched preclinical models for patients so that we're able to learn from their overall experience here, but also be much larger screens and build up a knowledge base where we may be able to take new drugs into the system going forward. And if we have a look at this as the results showing you here for about 140 patients, we can break the classes of patients that we would see as follows. The first is a group that we see to be clearly actionable. We see that about 2% of our patients are hypermutated, normally through MMR deficiency. They're showing you here the circle plots for the patterns of the genome damage. These are normally genomically stable but have thousands upon thousands of mutations, generating thousands of neoantigens, and indeed are ripe candidates for immunotherapy. We have 9% of patients that are BRCA deficient, either through germline or somatic events, and they can be recognised by their genomic instability, their signature 3 in these events. We also have about 9% of patients that in fact have no KRAS oncogene within them, which means there is some other oncogene in those tumours that is driving their disease. And they are gold for us because it may be that those particular oncogenes in their disease are druggable, where KRAS gain-of-function mutations are not. And indeed, if we go through and have a look at the likes of that BRCA deficiency segment, we showed some years ago that if we did studies of exceptional responders, so those patients that had advanced disease, went on to palliative care, got a particular agent, and their disease melted away, and they've been alive for many years afterwards from pancreatic cancer. We in fact recognise that these sorts of patients almost invariably had got a platinum-based agent and indeed had BRCA deficiency, which we were able to show through whole genome sequencing. More recently, there's been similar studies done showing that either platinum-based approaches or in fact PARP inhibitors can be very effective in these individuals, showing you that genomically we may be able to identify these patients and push them towards this. And indeed, if anyone was at ASCO this year, you would have seen some of the first good news that we've seen in pancreatic cancer, certainly in my time working on it. And when I started, I had lots of hair and was a young lad. And that is, we've effectively seen a doubling of progression-free survival in advanced pancreatic adenocarcinomas through the use of PARP inhibitors on those patients that are BRCA deficient. So we need to capture every one of these now going forward within our cohort. I should also say those KRAS wild-type patients, they're a really interesting bunch. They're not particularly common, but in our experiences when we dig in and have a look at them, we find at least half of them have oncogenic fusions that are seen in other carcinomas that are readily druggable. And indeed, there is now data coming out of case studies where these sorts of events can be treated. And indeed, there are other phenomena such as inframe BRAF and most recently described NRG fusions that have been seen in the German cohort that we're actively pursuing within these individuals. That means something like 10% of our patients right now, we need to be able to identify fusions and their mutation readout in order to actually have a chance of tackling that disease. So collectively, that group there, I should say, is about 20% of our patients. There is another set here that, as I mentioned, about 4% of our pancreatic cancer patients actually don't turn out to be pancreatic cancer. Remember that this study, many of our patients are actually presenting with advanced disease, and in the case of pancreatic cancer, you would normally go and biopsy their liver rather than their pancreas if they've got advanced disease. So we've got a presumption of pancreatic cancer will have a liver metastases, and indeed they'll do a limited amount of histology on that as best they can. And what we're finding for these particular tumours is that we need to look for what evidence can we build up as to are we certain they're pancreatic or not. And we've got 4% where we have very good evidence that they have not been pancreatic adenocarcinoma, and for the majority of them we've then been able to confirm that that was the case. And then finally we have a set of about 30% of patients where there is a number of druggable targets, but at this moment in time we can't currently test them with trials, but we're using the preclinical models to prepare those in to be involved in the next phase. Just finally, this idea of rare cancers. I know we talk about the term rare cancers make you think they're not very common. Collectively, rare cancers make up quite a significant proportion of our cancer patients each year. And because of the way that we've perfected the art of oncology, these patients are being left behind. So we've been very keen to go through and pursue these sorts of individuals to see what genomics can tell us. And indeed, what I'm just showing you here is 4 patients where the frequency is 2.5 per million, 1 per million, 5 per 100,000, 1 per 200,000. So in this case there, we've got a couple of neuroendocrine tumours, we've got a leiomyosarcoma, we've got a Ewing's, and just highlighting these for different reasons. These are rare cancers that come across us. First one was a diagnostic conundrum where we were able to show that ERG in fact was the fusion, but it wasn't recognised by traditional techniques because of the complexity of the fusion event. Here's a tumour here, it's an ovarian neuroendocrine tumour that in fact we're able to show is genomically unstable and a profound BRCA signature that went on to PARP inhibitors. Here's a leiomyosarcoma that surprisingly had a homozygous deletion of BRCA2. There's next to nothing known about this genomically. This patient has gone on and responded very positive to PARP inhibitors. And this individual here is an example of a very rare type of pancreatic cancer, which was KRAS wild type, but indeed it's found to have an oncogenic fusion, which then been able to go on to therapy. And really, we need to build these cases up one patient at a time. Finally, we know that all too often that in case of cancer patients, they may respond initially well to a treatment, and then over a period of time, their cancer may come back. This is high-grade serous ovarian cancer, a patient experience, where we can see that the recurrent use of drugs, ultimately the patient becomes resistant and passes. There are genomic and epigenetic events that are taking place in these patients. We've been very keen to go through these sorts of cases, trying to understand what we can see. And indeed, just showing you here, I've got 3 different neuroendocrine tumours. So these are both rare and relapse. And indeed, by the sequencing approaches, I've given you 3 examples of where we've gone ahead to change the course of their following treatment, either through the identification of new druggable targets, or indeed, in this case, the accumulation of mutations that are specifically involved in making you resistant to platinum-based therapy, which is the first treatment, and resistant to immunotherapy, which is the second treatment, but indeed recognises the potential of a third druggable target within this patient taking this approach. So I guess just finally, what I'm trying to describe to you is I think we're still very much at this early stage here, at the stage of pioneering genomic pathology, but really in order for us to keep going further, we need to have more information and more data coming in around patients and experiences. We're only going to be able to do that through a global-based approach in order to actually see this vision of precision oncology being realised. Just finally, I just really should say that this work stands on the shoulders of a lot of very, very clever people that I have the pleasure to present for. This team over here is largely the UMCCR activity group, and indeed I specifically want to highlight Oliver Hoffman. Indeed, none of this would be possible without our national collaborators and indeed our colleagues within the likes of the ICGC. And with that, I'm happy to take questions.